Okay, our next speaker is Sczyrba, who's also been deeply involved in the CAMI challenge.
Yeah, okay, thank you very much. Yeah, I will talk a bit about the assembly metrics again. Alice mentioned this already, but I want to go a little bit more into the detail. And actually, what we want to do is encourage you to come up with metrics, right? Even for binning or profiling tools, even for the tool Fernando just presented. As Alice said earlier, we want to involve the community. So if you have ideas about different metrics we could use actually for these evaluation, just come up and you can actually plug this into our system. If you have a sort of standardized kind of Docker container, it's easy to plug it in and just run it on the different results we got from the different tools and see if your metrics actually makes more sense than what we came up for metrics. So I want to talk a little bit about assembly metrics, which is not really which are not really new metrics, but I want to show you a little bit in more detail how it looks for the CAMI data sets. So again, you've seen this a couple of times now. I'm actually going to talk about the high complexity data set, which has about, 1,100 genomes and plasmids in it. And again, the way we generate the gold standard looks like this. So we start with an initial assembly of a known genome, right, of a unpublished but isolate genome, which of course most of the time is not a closed genome. So we start actually with multiple contigs. And the first step we do is we remove small contigs smaller than 1 kb. And from the remaining contigs, we simulate the reads. And whatever region has at least one X coverage will end up in the gold standard, right? And this is basically the perfect assembly you would get, which of course no assembler actually gives you, right? Because one X coverage is pretty low, but at least that would be the ultimate goal, what you want to get out of a data set. So this is a gold standard, and of course the number of gaps you get depends on the abundance of a genome. If a genome is not very abundant, you have more gaps because you have less reads. If it is very highly abundant, then you basically represent the complete genome. We take the results from the different assemblers and map it back to the genomes. That's the easiest thing you could do. And of course, okay, first of all, you can do some statistics which is independent of the reference. You can just count the number of contigs, look at the largest contig length, look at the total assembly length, look at the N50 and so on. These are standard metrics you usually look at when you do isolate assemblies, genome assemblies. But as we have the references available, we map everything back to the reference. We're actually using MetaQuast for this. And then you can calculate other statistics like this one here. So these are the contigs from the assembly. This is the reference. So by aligning them back, we can start with a genome fraction, like how much of a genome is actually covered, right? These red parts of the genomes here, and just look at the genome fraction. You can look at duplication ratios, for instance. So here there are two contigs which actually represent the same part of a genome, so it's duplicated. You can look at the number of gaps you get at your assembly. You can look at the largest alignment length, or you can look at the number of unaligned contigs. So contigs which actually didn't exist in your reference. And when you do this for the high complexity data sets for the different assemblies, it actually looks like this. This is showing the total assembly length for each of the reference genomes, actually. Right, so we map it back to the references, and each dot here is one genome, and it's overlaid actually by the box plot here. And when you look at these results for the different assemblers, and again, same color means it's the same tool, but run with different parameters. You already see some differences in the assemblies, and what you see is that, for instance, this tool here, Velvet, basically doesn't assemble anything. Same for Ray, right? So the total assembly length is pretty small compared to the other assemblers. Now you can look at the number of contigs, and of course there's some correlation between these two things. And you can look at the N50. And when you just look at the N50, I mean, there's not much information you can get out of it, at least not in a big picture. Of course, you see some assemblers that do a little bit better job, but overall, the N50 for metagenome assembly is pretty bad. You can look at misassemblies per megabase, for instance, right? And again, it doesn't look very different for the different assemblers. So some tools are doing a pretty good job, like Velvet actually. There are not much misassemblies per megabase, but the problem is there's not much assembly either. So you want to combine all these different metrics to some sort of score. That would be the ultimate goal, actually. So for presenting the different results, we actually mostly stick with the genome fraction because we think this is actually what you want to get out of it. You want to get most, or at most of each of the genome as possible, right? And this is how this looks like. You've seen this plot before. And again, you see like the median genome fraction recovery is pretty good for some assemblers, and these are actually the assemblers that use multiple k-mers, like iterating over multiple k-mers, and the ones which only use a single k-mer are not performing very well. And again, you've seen this before. Let me just summarize this again. We made we basically split it into two different groups: the unique strains meaning there's not any genome in the metagenome which has an average nucleotide identity above 95%. So they should be easy to assemble because there is not much like cross matching between different genomes. And then we have the common strains, which are strains where the average nucleotide identity is larger than 95%. And as Alice pointed out earlier, like all of the assemblers have problems with these strains. And this is what you basically would expect, because whenever they become too similar, they are either going to be just one sort of consensus genome, right? If it's like a 99% identical, it will just be collapsed into one genome. Or you have that many errors that it actually the assembly breaks apart completely because you have so many what looks like errors to the assembler that the contigs are actually getting smaller and smaller and smaller, and then at some point it's below the minimal cutoff for the contig length, and then you don't see the genomes anymore. And so this is actually why we plotted it this way, because then at least you get an idea what's going on, which you don't get from these sort of global box plots. And so this is a little bit more detail here about the different assemblers. This is actually, let me see, this is Ray up here. This is a Ray assembly with just one k-mer. This is, I don't know, I cannot read it anymore. This is a different assembler, but also using a single k-mer. And what you see is basically using a single k-mer targets the assembly at a specific coverage, right? So down here is the coverage of a genome. Each dot is one reference genome. And we have different abundances, which goes from 0 to like 1000x sequencing coverage in this case. So of course you don't assemble anything with very low abundance. The k-mer basically targets a particular, let's say, coverage range, right? And if you only use a single k-mer for your assembly, then basically you only get part of the metagenome assembled. And so this is the same assembler. This is Velour, changing the parameters a little bit and actually going from a k-mer size of 31 here all the way up to like 63. And what you see is you see the shift basically. With lower k-mer, smaller k-mers basically assembles the low abundant genomes. Larger k-mers assemble the high abundant genomes, right? And as you can imagine, when you try to combine this, when you do some sort of iterative way of assembly, which is what the best performing assemblers are doing, they start with small k-mers, they iterate across a range of k-mers from, let's say, 21 to 120 or something like this, and then you basically assemble the whole range of your abundance. But Apparently, some assemblers have some weird problems, like this one here. This is Minia. The early version of Minia has problems with coverage around 100x, but when it's more coverage, and it's actually doing a pretty good job. This one here doesn't assemble anything with very high coverage, right? So you can see there's no perfect assembler out there, basically, and the main challenge for the assembler are the common strains, and these are the blue colors here. So whenever you have closely related genomes in your metagenome, they are basically lost. You cannot see them in the assembly at the end. Okay, so that's it from my part. This is just, again, showing you some metrics we use for the assembly. And again, if you think there are better ways to do it, but maybe by looking at graphs of the assembly and then sort of evaluating the assembly graphs instead of the contigs you get out of it, just come up with your ideas. And tomorrow we have a session where we can actually discuss the ideas and hopefully implement this in the pipeline. Okay, thank you very much.
Thank you very much, Alex. Do we have questions? We now finally have time for several questions if you're feeling chatty.
Okay, if there's time this time, I'll come back to my earlier question on exactly that.
Mm-hmm.
So I liked your description of the challenge of assembling strains. If thinking about it as a continuum, if you have two strains that are identical, of course that'll be one genome. As they differ by more and more and more loci, the assembly graph will, as Adam was just saying, kind of bubble up until then it melts into two different assemblies at an extreme, and then you can evaluate it like isolate genomes.
Exactly.
In that intermediate range, so we see a lot of multiple strains using reference-based approaches where you map to some reference, call SNPs. There's presumably a world in which you could evaluate the assemblies like that by considering each cluster of related strains to have one reference assembly, and then evaluating in some way, once you map back to the new assemblies, what does that tell you about the expected SNP profile? I think there's also a world, or maybe a complementary evaluation, where you could evaluate the assembly graph directly and have an idealized assembly graph, maybe containing a lot of bubbliness, and then the challenge for the metagenome assemblers to recapitulate the correct bubbles in that case.
Mm-hmm.
Do either of those seem like what you see?
Yeah, it makes sense. It definitely makes sense. I mean, I think it's still hard to evaluate. I'm not really sure how to do the evaluation, but maybe it is useful for the developers of the assemblers also, right? So because you could look at the graph first to see if the graph is actually correct. Because the next challenge is how to make up the genomes from the graph, right?
Yeah.
Because that's a challenge in itself. Because you might have all the variants in your assembly graph, but then at some point you want to get the FASTA sequence out of it.
Well, except maybe you don't.
Or maybe you don't. Maybe you just want to keep the graph.
If you have two strains that are almost identical, you may or may not have any way to create two FASTA files for those. You may have one FASTA file that's the reference genome, and then some way of assigning variants to loci across it.
Yeah, that's true. I mean, it's basically a phasing problem which you cannot solve for the graph with the short reads.
Yeah.
But maybe the long reads will help.
I think it's going to be harder to evaluate with long reads because assemblers tend to smash more, and you don't have the nice Brown framework of Cambridge operating.
Yeah,
Overlaps gets much messier, and I think it'll be harder to evaluate.
Yeah, we'll see. So, well, we really have to think about it for the next challenge because we are including the long reads, and then we see how to evaluate.
If you want to talk about mapping and variant calling, we do a lot of that. So I'd be curious to talk about that. Assembly, I don't actually know anything.
Okay.
So.
Okay, thanks.